Hello everyone, I'm Anshu Gupta. I'm a second year PG student in CSE department at UC San Diego, and now I'll be going to present my work on accurate, scalable and fully automated inference of species tree from raw genomic assemblies using ROADIES. Let's start with a brief introduction and motivation behind our work. Now we are in the era of phylogenomics, where genomic data is widely being used to construct the species tree. There has been a significant improvement in the sequencing technologies in recent years, and that has led to a huge explosion of the number of genomic data available these days. As we can see in this graph, the data growth is almost exponential in several life forms, be it mammals, fishes, and birds. Now, with this improvement in sequencing technologies, there are multiple consortium which aims to sequence tens of thousands to even millions of species on Earth. And this wealth of data has immense potential to give us a better picture of how species would have evolved over time, and also helps us settling a lot of debates that is related to the complex branching patterns within the tree of life. To give you an example of one such debate, let's take a look at the latest avian phylogeny. So this picture shows the very latest phylogeny of 363 different species of birds. This gives us a latest picture of how birds would have evolved. Now, if we just look back 10 years from now, there was another paper which proposed the phylogeny of 40 different species of bird. Now, if we look closely, both the trees at the order level had some differences. Interestingly, both the trees had multiple overlapping authors. That means even the authors revised the initial assessment of the correct tree once they had more data and better bioinformatic tools over last 10 years. However, researchers are still not 100% sure with some parts of the tree yet. That means even after 2 centuries of studying the phylogeny, we are not at the position of correctly estimating the species tree. Now I'll talk about how the species tree are generated conventionally and what are the methods being followed. There are two main approaches in estimating the species tree. The first approach is doing multiple whole genome alignments with the entire genomic data. Now, multiple whole genome alignments are computationally very expensive. Hence, it's not a feasible method to follow, especially if you have thousands of genomic data available these days. Also, it requires guide tree as an input. So that means for the species tree to be accurate, you need to have your multiple sequence alignment to be accurate, and for that, your guide tree needs to be accurate. And that is what we are trying to estimate. Hence, this becomes kind of like a chicken and egg problem, and that's why this process is erroneous. Now, another method which is mostly followed in conventional approaches is orthology-based species tree estimation. This requires us to first annotate the genes, establish the orthologs, and then create the gene trees and the species tree. Now, gene annotations are essential because they help establishing the orthologs easily. Orthologous sequences, they share a common ancestor through speciation. Hence, researchers specifically focus on picking out orthology because that tells us more about the speciation events and how species evolved and share their similar functionalities. After finding the orthologous sequences, then they do the gene tree estimation and the species tree estimation. Here, most existing works focused on only single copy gene trees, which are reconstructed from the set of orthologous genes. Also, these gene tree estimation and species tree estimation, why it has been done in two ways? Because of the presence of gene tree discordances, where different genes in a genome can have different evolutionary histories. Now, to get rid of that, there are various discordance aware tools available these days. One such example of discordance aware tool is AstralPro, which is widely adopted and being used in multiple bioinformatic pipelines. AstralPro estimates species tree from the list of gene trees given as an input. It follows a homology-based species tree estimation. That means it does not require user to separate out orthologs and paralogs, thus giving a unique opportunity to automate the process and remove these manual steps. Now, there are certain challenges in the second approach. First, correctly distinguishing orthology and paralogy is challenging because they may look familiar, they may look similar because they share a common ancestry, but all of them are not due to speciation. There are some duplicate copies of the genes as well. Hence, it's challenging to find true orthology. The second challenge is to manually annotate the genes because that requires some sort of manual tuning, domain expertise to perform it correctly. And that is why the errors at this step can propagate throughout the entire pipeline and making the entire process error prone. And because of these hindrances, there has not been much attempts to our knowledge in automating the entire process of estimating species tree end to end. There are recent developments of discordance survey tool, as we saw in AstralPro, and that removed the requirement of separating out orthologs and paralogs. But even with that, the individual stages of the conventional approaches we see are being done individually, which is non-feasible if we are going to follow it for tens of thousands of genomic data in near future. Hence, it's becoming very crucial to have an automated method to reliably generate the species tree which matches the evolutionary history the most, and also catering to these large-scale datasets. And with that, our objective of this work is to develop a reference-free method where we do not have any reference bias, an orthology-free method where we don't want user to separate out orthologs and paralogs, an alignment-free method where we do not want any whole genome alignments, and also to be discordance aware to generate a species tree from the genomic data. And this is how we named our tool ROADIES, which fulfills all of these objectives as highlighted. Now, ROADIES is an automated tool which creates a species tree directly from the raw genomic assemblies. Along with the species tree, it also generates gene trees. Now, how ROADIES solves existing challenges? There are certain things that we do differently than rest of the conventional methods. Overall, we follow the approach of homology-based species tree estimation. Here, we first randomly sample the genes instead of manually annotating the genes. Now, I want to mention that when we say gene, the term gene here referred to as C genes or coalescent genes, which are short recombination-free regions, and not the protein coding genes, which is being considered in most of the conventional methods. So there are multiple benefits of doing random sampling. First, here we are not just limiting to protein coding genes, but we also sample intergenic regions because intergenic regions follow the assumption of neutral evolution better. Secondly, along with this random sampling, we eliminate the reference bias since genes are sampled randomly with uniform distribution across all genomes. Third, we do not need to do any sort of multiple whole genome alignments, which eventually requires guide tree as an input. Apart from the random sampling, we do homology-based inference instead of orthology inference, because in orthology inference, user need to specifically pick out orthologs and paralogs, but homology inference is much easier than that, and that allows to make our tool much more faster. Apart from this, we do homology-based species tree estimation instead of orthology-based species tree estimation, and also being discordance aware by taking the advantage of AstralPro. And these differences with respect to the conventional approaches simplifies the computational process and enables us to automate the entire pipeline end to end. Thus, ROADIES become much more faster and less error prone without compromising on accuracy. So the entire pipeline of ROADIES is divided into 6 different steps as shown here. First, we randomly sample the genes from the input genomic assemblies. Second, we use whole genome aligner to take sampled genes and find where the homologs are using this pairwise sequence alignment tool LAZ. Then, we discard the bad matches and also repetitive matches from a small genomic region. Then, we align the homologous matches of each of the genes individually using this multiple whole genome alignment, multiple sequence alignment tool called PASTA. Once we get this multiple sequence alignment for each of the genes, we then estimate the gene trees of each of them using this gene tree estimation tool RAXML-NG. Note that some species may have multiple homologs detected and some none at all. Finally, once we have this list of gene trees, we then estimate the final species tree using AstralPro as discordance aware technique. So this sums up the entire pipeline of ROADIES and shows how it actually automates and estimates the final tree directly from the input sequences. Now there's one problem with whatever we do. We don't know how many genes to sample. So for some data sets, it's very simple. Like for mammals, we can even get the optimal tree with 4,000 genes. But for birds, which has high level of discordances because it evolved quite rapidly, we may even require 60,000 to even 100,000 genes to finally get the right species tree. Now, how to know how many genes are optimal for which kind of data set? So for that, we came up with an adaptive algorithm that basically takes this tree confidence scores into account. So it iterates the entire pipeline of ROADIES multiple times, and at every iteration it keeps track of the final tree confidence. And if the tree is confident, it will stop. Otherwise, it will keep going further and further with more and more gene trees. So the idea is here that to get the confident tree with the right number of genes for any kind of data sets. So now let's talk about the ways in which we evaluated our results. We picked 3 different data sets, mammals, flies, and birds, and estimated their species tree. We then evaluated the correctness of this tree by comparing it from the existing papers, which is widely approved by the community, and we considered that tree to be our reference tree. So then we evaluated the differences between the ROADIES tree and the reference tree using normalized RF distance as the distance metric. The lesser the distance is, the more accurate the tree is compared to the reference. We measured this normalized RF distance in 2 levels, species level and at the order level. Along with the tree distances, we also measured the confidence of the tree wherever the branches were different from the reference using the local posterior probability and the quadrat scores. Now, because of short of time, I'll only show the results with mammal datasets. So this is the final species tree that we got from the mammalian datasets. Now, I understand it's quite hard to view the details here, but I would like to specifically emphasize that ROADIES was able to group the species in its right phylogenetic orders, similar to what Zoonomia proposed. In addition, we also see the green colored and blue colored graph that basically depicts how many genes were selected from each of the species in the beginning and also how many genes were eventually aligned with each of the species. Now, this figure shows the order level comparison between the ROADIES tree at the right hand side and the reference tree at the left hand side. And as you can see closely, both the trees are highly similar to each other. But there are minor differences which is highlighted over here. Interestingly, some research analysis supported our results, some supported the results which is proposed by the reference tree, and some proposed a different alternative. This means even scientists also did not reach to a common conclusion of the correct phylogeny of these highlighted orders. But more or less, ROADIES was able to accurately estimate the relationships for the well-settled orders with lesser debates. Now these are the plots which is generated by the ROADIES using the convergence mechanism. In the first plot, we can see how the confidence of the final tree increases with more and more gene trees unless and until it gets saturated where our convergence algorithm stops. In the last plot, we can also see how the normalized RF distance decreases with more and more gene trees at both the levels. That means it becomes more and more closer to the reference tree. That means it becomes more accurate. We did the similar type of analysis with flies dataset and we got accurate results here. Same we did with birds dataset, except some highly contested branches. Now, apart from the accuracy of the final trees, we also evaluated the speed up compared to the conventional methods. Now, to evaluate the speed up, we measured the wall clock time of the entire ROADIES pipeline from start to end for the mammals dataset, and also we found the total runtime of the state-of-the-art tool TOGA, which infers the orthologs from the annotated data. So we found that the total wall clock time taken by TOGA was around 176 times higher than the ROADIES. And here we just compare the entire ROADIES runtime from end to end with only 2 stages of the conventional method. So this shows how fast our tool is. We also tested the scalability of ROADIES with increasing number of system cores and also the increasing number of species. We observed that ROADIES scales super linearly but less than quadratically with more number of species. With more number of cores also, we saw a runtime decrease of the ROADIES pipeline significantly. But with higher cores, like especially with 256 cores, we saw a slight increase in the runtime, and that can cause because of an increase in the communication overhead over the computational efficiency. And these results are taken from single node multiple core AWS instances. For better scalability, we are currently planning to scale it further by applying multi-node parallelization so that ROADIES can run over huge clusters. Now, our long-term aim for ROADIES is to support the datasets from diverse timescales. So far, we found impressive results with like mammals, flies, and birds. But now we are aiming to run ROADIES with fish datasets, which evolved around 500 million years ago, which is much earlier than the datasets we used. On the flip side, We also would like to span ROADIES for estimating the phylogeny of SARS-CoV-2, which evolved just like few years ago. So that means we would like to extend our work to support both sets of data sets from diverse evolutionary timescales. So that pretty much sums up my work. So in today's talk, we saw how important it is to construct an accurate tree for thousands of species and how conventional approaches are not just feasible to continue in near future. Also, scientists are still debating on the correct notion of the tree. ROADIES solves all these existing challenges by automating the entire pipeline of the species tree estimation, and also it's highly configurable to the user requirements. It's highly scalable. When we compared with mammals dataset, it was around 176 times faster and also gave accurate results for mammals, flies, and birds dataset. With that, I'd like to acknowledge my advisors and collaborators, especially my advisor Yatish Turakhia and Mirarab for their constant help and support. ROADIES is now submitted to bioRxiv and the preprint is available online. The code and documentation is also available online, so please feel free to check it out, run it for your datasets and provide your feedback. Thank you everyone.